Students, now we are going to revise what are the things that we have covered so far in mutations. Mutations are broadly classified into three types. One is gene mutation. This is one type of mutation. Gene mutation. The second type of mutation is genomatic mutation. Genomatic mutation. And the third type of mutation is chromosomal mutation. So mutations are broadly classified into three types: gene mutation, genomatic mutation, and chromosomal mutations. So for better understanding of what is gene mutation, we must be in a position to recall what are the purine bases and what are pyrimidine bases. So for better understanding of gene mutation, we should have which are purine bases and which are pyrimidine bases. The purine bases are alanine and guanine. The purine bases are alanine and guanine, whereas the pyrimidine bases are thymine, and cytosine. These nitrogen bases, they form respective nucleotides. So you should remember what are purine bases and what are pyrimidine bases for better understanding of gene mutation. Then what is a gene mutation? Mutation means change. It is change in the structure and composition of gene. That is gene mutation. A gene mutation is a change in the structure and composition of the gene, and a gene mutation is also called point mutation. The other name of gene mutation is point mutation. And this gene mutation or point mutation is classified into two types. It is classified into two types. One is the gene mutation by substitution and gene mutation by frame shift mutation. So the gene mutations are of two types. One is gene mutation by substitution and gene mutation by frame shift mutation. By frame shift. And what is substitution? And what is frame shift mutation? What is substitution and what is frame shift mutation? Substitution means one nucleotide or nitrogen base. These are all nitrogen bases: alanine, guanine, thymine, cytosine. Or they form their respective nucleotides. One nitrogen base or nucleotide is replaced by the other. That is substitution. So what is a substitution? Substitution means one nitrogen base nitrogen base or its nucleotide is replaced by the other. Say for example, one purine base is replaced with the other purine base. One purine base is replaced with the other purine base. Adenine is replaced with guanine or guanine with adenine. Or one pyrimidine base is replaced with the other. That is, thymine is replaced with the cytosine or cytosine by thymine. Substitution is one kind of gene mutation wherein one nitrogen base or its nucleotide is replaced by the other. When a purine base is replaced by another purine base, when a purine base is replaced by another purine base. Now let us examine this. When a purine base is replaced by another purine base. Now I will write G T T GTT as a codon. A codon is one which has three nitrogen bases or its three nucleotides. One is guanine, thymine, thymine. All the three nitrogen bases or all its nucleotides form a codon. Now observe the change. This GTT, a codon, it undergoes mutation and forms ATT. So GTT becoming ATT. What is G? G is guanine. Guanine is a kind of purine. G represents guanine. Guanine is a kind of purine. It is a kind of purine. Now this GTT becomes ATT upon mutation. That means the guanine is found to be replaced with adenine. Guanine is found to be replaced with adenine. That means one purine base is replaced with another purine base. One purine base is replaced with another purine base. Now we will examine one more. We are going to examine one more. Now you look at this example. This is a codon CAG, a codon with three nucleotides or three nitrogen bases, cytosine, adenine, guanine. This upon mutation becomes Cytosine is a kind of pyrimidine. This is replaced by another pyrimidine, that is thymine. Now in this first example, a purine is replaced by another purine. A purine is replaced by another purine. In this case, a pyrimidine, that is a cytosine, is replaced with another pyrimidine. When purine is replaced by another purine or pyrimidine by another pyrimidine, then such a substitution is called transition. Try to remember this. It is called transition. Students, try to remember. This area is very, very important from the NEET point of view. They may give any codon and they may ask you what is the type of mutation that the codon has undergone. If this is given, the GTT codon upon mutation forms ATP, then what is the type of mutation that is involved means it is substitution by transition. It is substitution, of course. Under substitution, it is a transition. When one purine base is replaced by another purine base, or one pyrimidine base by another pyrimidine base, then the substitution is called transition. Then the substitution is called transition. Hope you have understood what is transition. When one purine base is replaced by another purine or one pyrimidine by another pyrimidine, then the mutation is transition. Transition is one kind of substitution. It is one kind of substitution. Now let us examine the other case. Now let us examine the other case. Now I will write a codon. The codon is ATT. This is the codon. And this codon has undergone mutation, a change, and resulted in a codon called TTT. The codon is ATT, which has undergone mutation and forms TTT. If we examine this change, we can understand that a purine base, that is adenine, is replaced with a pyrimidine base, that is thymine. So, in this case, purine is replaced In this particular example, adenine is a purine base which is replaced with the thymine, that is a pyrimidine base. A purine is replaced with pyrimidine. Sometimes a pyrimidine is replaced with purine. Say for example, there is a colon CAG, which upon mutation forms GAG. In this particular case, cytosine is a pyrimidine. The cytosine, that is a pyrimidine, is replaced with a purine base, that is guanine. CAG becoming GAG. Now try to compare this case with that case. In transition, one purine base is replaced by another purine, or one pyrimidine by another pyrimidine. In transition, one purine is replaced by another purine base, or one pyrimidine by another pyrimidine. That is a transition. In these particular cases, one purine is replaced with a pyrimidine, or a pyrimidine with a purine. Then it is also a kind of substitution by transversion. Substitution by transversion. So you must thorough with the basics, which are the purine bases of DNA and which are the pyrimidine bases of DNA. Now you have to understand what type of gene mutation that the given codon has undergone. Whether it is one purine replaced with another purine, or pyrimidine with another pyrimidine, then it is a case of substitution by transition. When one purine is replaced with another pyrimidine, or pyrimidine by a purine, then it is a substitution by transversion. I hope you have understood and you try to remember this. Such questions will likely to appear in NEET examinations. Please try to remember. Okay. Gene mutation, I said it is of two types. One is substitution, gene mutation by substitution. Substitution may be by transition or transversion. What is this frame shift mutation? Let us try to understand what is a frame shift mutation. Frame shift mutation means to try to understand from, suppose this is the frame. Imagine that this is the frame. My palm is the frame. Now I will move the palm. This is shifting of the frame. Shifting of the frame. Now I move the palm, one of my palms. You can notice there is increase in the length of the frame. Increase in the length of the frame. This increase in the length of the frame is possible upon the addition of nucleotides. Upon the addition of nucleotides. Suppose this is the frame now existing. Upon the deletion or removal of nucleotides, now again there is shift in the frame, decreasing in the length of the frame. So addition of nucleotides, one or more nucleotides increase the length of the frame, and deletion of one or more nucleotides decrease the length of the frame. Then it is called frame shift mutations. So in frame shift mutations, there is addition or deletion of one or more nucleotides. One or more nucleotides. So, what is a frame shift mutation? Frame shift mutation due to loss of one or more nucleotides from the frame, from the given frame. When there is a loss of one or more nucleotides from the frame, then the length of the frame decreases. Then the length of the frame decreases. Then the nature of the codons, also get changed. Let us examine. First we have a frame, for example, there is a frame called ATTGCC. This is the one frame available. ATTGCC GAG. Suppose this is the frame available. I am going to add one nucleotide. I may select a purine nucleotide or a pyrimidine nucleotide. I insert this nucleotide at any point in this frame. I insert this nucleotide at any point in this frame. Say for example, I have selected the nucleotide G, that is a guanine nucleotide, and its insertion at this particular point. At this point. There is no hard and fast rule that it should be inserted always at this point. So I have taken the nucleotide guanine nucleotide and its insertion at this particular point. So what is the change that we are going to notice? It is ATT. I have inserted the guanine here. So this is the newly inserted one, followed by G, C, C, G, A, G. Earlier, that is before mutation, there are 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 nucleotides. 9 nucleotides before mutation. 9 nucleotides with 3 codons. This is the one codon. This is another codon. This is another codon. One is ATT codon, GCC codon, and GAG codon. I have inserted a nucleotide G at a particular point, that is at this place. Now, how many nucleotides are present in this frame? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Earlier, there are 9 nucleotides. Now, one nucleotide is added, and it increases the length of the frame. Now, observe the codons, nature of codons. In this case, the first codon remains the same. It is ATT earlier. Earlier it is ATT, now it is also ATT. The second codon after mutation is GGC. Before mutation it is GCC. Is it not a change in the nature of the second codon in this case? There is a change in the nature of the second codon in this case. The change in the second codon, in the nature of second codon, may alter the amino acid that it is going to code, that it is going to decode. Each codon represents an amino acid. Earlier it is GCC, it may represent one amino acid. After mutation, it is no more GCC, it is GGC. It may alter the amino acid that is it going to decode. Then the third codon is GAG. Here, what is the third codon? It is CGA. Again, the third codon gets altered, gets mutated. It may even change the amino acid. May change the amino acid. And one codon is left free in this case. You observe this. The addition of one nucleotide alters the fate of the protein chain that is going to be synthesized. The addition of one or two nucleotides may alter the fate of the protein chain that is going to be synthesized, or may alter the character that is going to be derived. Therefore, here in this case, there is addition of one or more nucleotides to the frame, to to the frame. Addition of one or more nucleotides to the frame. It is not loss, it is addition. Addition of one or more nucleotides to the frame. Increases the length of the frame. This is called a frame shift mutation by addition or insertion. It is called frame shift mutation by addition or insertion. So in this particular case, we have examined the insertion of a nucleotide, that is a guanine nucleotide, at this point. As a result, the length of the frame gets increased. This is frame shift mutation due to addition or insertion. Now, let us examine the second case. Now, this is the frame that we have, that is ATT, GCC, GAG frame. Now, it has undergone mutation and resulted in AT, D, CC, G, A. Now let us examine this. When you examine this, before mutation, this frame has 1, 2, 3, 4, 5, 6, 7, 8, 9 nucleotides or 3 codons. 3 nucleotides constitute 1 codon. Now, count the number of nucleotides after mutation. 1, 2, 3, 4, 5, 6, 7, 8. Here there are 9 nucleotides. Here there are 8 nucleotides. That means there is loss of 1, of course in this case, or more nucleotides, from the frame. Then what is the consequence of it? Earlier, that is before mutation, we have ATT as a codon. Now, the starting codon is ATG, not ATT. The second codon is GCC. Here, the second codon is CCG. And the third codon is GAG. And here the third codon is incomplete. It is deficient by 1 nucleotide. That means in this particular case, there is decrease in the length of the frame. In this particular case, there is increase in the length of the frame upon the addition of 1 or more nucleotides. Then it is addition or insertion. Here, there is a decrease in the length of the frame upon the removal of 1 or more nucleotides. What is the nucleotide that is removed in this case? The thymine nucleotide is removed. The thymine nucleotide is removed in this case. Therefore, it resulted in a decrease in the length of the frame mutation. The earlier case is a frame shift mutation due to addition. And this frame shift mutation due to addition of one or more nucleotides or due to deletion of one or more nucleotides may alter the nature of codons, may alter the nature of the amino acids decoded by each codon, may change the structure of a protein resulting in a new character. And this frame shift mutation is also called gibberish mutation. It is also called gibberish mutation. The frame shift mutation is also called gibberish mutation. So, the gene mutation is of two types. One is a substitution and the other is frame shift mutation. Substitution may be by transition or transversion, and frame shift mutation or gibberish mutation is by addition or by deletion. Addition or by deletion. Okay, students, now we are going to discuss the second type of mutations. The second type of mutations are genomatic mutations. Genomatic mutations. In gene mutation, we have come to learn that the nature and the composition of the gene get altered. But in genomatic mutation, there is change in the number of chromosomes. Mutation which results in the change in the number of chromosomes is genomatic mutations. So, a mutation which results in the change in the number of chromosomes. Then it is called genomatic mutations. is genomatic mutation. And this genomatic mutation can be studied under two heads. One is euploidy, and the other is aneuploidy. The genomatic mutations can be studied under two heads. One is the euploidy and the other is aneuploidy. Then what is the euploidy? Euploidy is a kind of genomatic mutation. Then what is the euploidy? It is a kind of genomatic mutation. In euploidy, there is a change in n set of chromosomes. There is a change by n set of chromosomes. There is change by n set of chromosomes. Then it is euploidy. If a cell has n set of chromosomes, it is haploid. If a cell has 2n set of chromosomes, then it is diploid. If a cell has 3n set of chromosomes, then it is triploid. Change in the chromosomal number by n set of chromosomes. Say for example, cell is diploid. There is a change in the number of chromosomes by n set, that is plus n, 2n plus n. Then 2n plus n gives rise to 3n condition. 2n plus n is equal to 3n condition. That is a triploid. Say for example, there is a diploid condition initially. There is change in chromosomal number by n set twice. By n set twice. N set plus n set. I said n set twice. It results in 2n plus n plus n. That is a 4n condition. That is a tetraploid. Then there is initially diploid condition. There is a change in the number of chromosomes by n set 3 times. That is 2n plus n plus n plus n. That is equal to 5n. That is a pentaploid. That means the change in the number of chromosomes, that is a 2n set of chromosomes, by n set of chromosomes is euploidy. Such euploidy type of genomatic mutation may result in triploid condition or tetraploid condition or pentaploid conditions, hexaploid conditions, heptaploid, that is 7n condition, or octaploid condition, nonaploid, 9n condition, decaploid conditions. That means from 3n set, 3n condition to decaploid, 10n condition and so on, it is a polyploid condition. So, the occurrence of polyploid condition is a case of genomatic mutation. So, occurrence of triploid, tetraploid, pentaploids, genomatic mutation, that is change in the number of chromosomes by aneuploidy. In this case also, there is a change in the number of chromosomes, not by M set, not by M set. How will be the change in the number of chromosomes over the diploid? That means it is a 2n plus or minus 1 or 2n set plus or minus 2. It is a 2n set, that is the diploid condition. You try to remember, in both the cases we are working over the diploid set of chromosomes. That you have to remember. Whether it is euploidy or aneuploidy, we are working over the diploid set of chromosomes. In aneuploidy, there is change in the number of chromosomes, not by N set, by one addition of one chromosome or deletion of one chromosome, or addition of two chromosomes or deletion of two chromosomes. Say for example, if it is 2n plus 1, then the condition is called trisomy. Trisomic condition. Trisomy. It is a 2n plus 1. If it is 2n minus 1, 2n minus 1, then the condition is called monosomy. Monosomy. Then the condition is called monosomy. If it is a 2n plus 2, 2n plus 2, then the condition is called tetrasomy or tetrasomic condition. What about this tetraploid? Tetraploid is the result of euploidy. Tetrasomic condition is the result of aneuploidy. Don't confuse. Triploid is the result of euploidy. Trisomic is the result of aneuploidy. So 2n plus 2 is tetrasomy. If it is 2n minus 2, 2n minus 2, it is called nullisomy or nullisomic condition. So the existence of trisomic, monosomic, tetrasomic, nullisomic individuals in the population indicates genomatic mutations due to aneuploidy. This is a very, very important point that you have to remember for NEET examination. The existence of trisomic, monosomic, tetrasomic, nullisomic conditions in the given population are the examples of genomatic mutations by aneuploidy. You will come across so many examples in genetics, in genetical disorders. For example, in a person, male, with a syndrome called Klinefelter's syndrome. The name of the syndrome is Klinefelter's syndrome. This Klinefelter's syndrome person has an extra X chromosome. He has an extra X chromosome. So this Klinefelter male person, instead of having one X and one Y chromosome, has one extra X chromosome, that is XXY condition. That means Klinefelter's syndrome person has 47 chromosomes. Usually the diploid number, 2n number of chromosomes in human being is 46. He has one extra X chromosome. Therefore, Klinefelter's person existence in the population is because of genomatic mutation due to aneuploidy. It is due to genomatic mutation due to aneuploidy. So, try to remember, these are very, very, very important. I have given only one example for your understanding. They may take any examples. Say, for example, Turner's female. There is another syndrome called Turner's female. In Turner's female, there is one X chromosome less. That is, it is a 2n minus 1 condition. That is, the Turner's female, instead of 46 chromosomes, have 45 chromosomes, and it is the result of the deficient of one X chromosome. So, Turner's female is an example of monosomic condition. Therefore, the existence of Klinefelter's male, Turner's female in the population are the examples of the genomatic mutations due to aneuploidy. Very, very, very important. Remember, these are very, very important. At the same time, we should spare some time. Sometimes we should think properly to choose the right answer. Therefore, the genomatic mutations which result in the change in the ploidy of chromosomes can be studied under two heads. One is euploidy and the other is aneuploidy. Now the third type of mutations are chromosomal mutations. In this type of mutations, there is a change in the karyotype of chromosome, that is morphology of chromosome. There is change in karyotype or morphology of chromosome. Karyotype refers to morphology. Morphology means structure. There is a let us examine the cases. Now, imagine this is a chromosome with a centromere and this is a chromatid. This is the arm of the chromosome. Of course, a chromosome has two chromatids. We have represented one chromatid and this is another chromatid. This is another chromatid. Now this is the chromosome before mutation. You just observe the diagram. This is the chromatid of the chromosome. This is one chromatid and this is another chromatid. Imagine this chromosome appears to be V-shaped. It has undergone mutation. Now, you try to observe the change that has happened. Now, what is the change that you have observed in this case? A part of the chromatid is lost. A part of the chromatid is lost. So, in this particular case, a part of the chromatid of a chromosome is lost. That means a V-shaped chromosome, upon the loss of a segment of its chromatid, becomes roughly L-shaped. That means there is change in its structure or morphology. There is a change in its structure or morphology. It is chromosomal mutation by deletion. It is a chromosomal mutation by deletion. This is one type of chromosomal mutation by deletion. Now observe the second case. Now, before mutation, we have this chromosome which is L-shaped. Now, after mutation, you observe the change. This is the centromere. Of course, this is the chromatid. And this is the chromatid before the mutation. Now, this is the chromatid after the mutation. So, what is the change that is observed? A part of the chromatid is added. In this earlier case, a part of the chromatid is removed. In this case, a part of the chromatid is added, or a new segment of chromatid is added. As a result, the L-shaped chromosome becomes V-shaped. The L-shaped chromosome becomes V-shaped. The earlier case is a deletion. The next case is addition. The addition of a segment of a chromatid. The addition of a segment of chromatid to one of the chromatids. Change the morphology of the chromosome. So the chromosomal mutation in the first case is by deletion. The this is a chromosome. Imagine that this is a chromosome with the segments A, B, C, D. Let this is the centromere E, F, G, H. Imagine that this is a chromosome with a centromere. This is the centromere of the chromosome with different segments, A segment, B segment, C, D, E, F, G, H. Now this is another chromosome. This is another chromosome with the segments, A, B, C, D, centromere, E, F, G, H. This is before mutation. Now mutation has taken place. Now mutation has taken place. Now notice the difference. Now this is the chromosome and this is the chromosome. Now watch the difference. A, B, C, D, E, F, G, H. A, B, C, D, E, F, G, H. Now these are the two chromosomes and different fragments of the two chromosomes. This is the before mutation and this is after mutation. Please see, the mutation may happen at any point. It may happen at any point, at any part of the chromosome. In this particular case, I have shown the mutation at this particular area. Watch this. This is the area where mutation has taken place. What will happen? These segments, that is BC, B capital B and capital C, are from this chromosome. And in this particular chromosome, the small b and small c segments are from this chromosome. That means there is mutation due to translocation. This is called mutation due to translocation. Exchange of segments of chromosome. Exchange of segments between two chromosomes is translocation. Exchange of segments. Exchange of the segments. These the segments B and C by this portion of this chromosome. And these segments, small b, small c, they are taken by this portion. That means they are mutually exchanged. The exchange of the exchange of segments between the two chromosomes is called translocation. So translocation is a kind of chromosomal mutation. It is also a kind of chromosomal mutation. The chromosomal mutation is due to deletion. The chromosomal mutation is due to addition. The chromosomal mutation is due to translocation. In translocation, there is exchange of segments between two chromosomes. Remember, that is translocation. Now we are going to observe another type of chromosomal mutation. Another type of chromosomal mutation. That is, now observe the change. This is the chromosome that we have with A, B, C, D. This is centromere, E, F, G, H. Suppose this is the chromosome with different segments. Now it has undergone mutation. It has undergone mutation in two different ways. Observe the first way of mutation. A, C, B, D, E, F, H, G. Now observe this case, second case. A, B, C, G, D, F, G, H. Now observe the chromosome before mutation and observe the chromosome after mutation. Here, the segments are rearranged. There is rearrangement of segments within the chromosome, within a chromosome. But in the earlier case called translocation, there is exchange of segments between the two chromosomes. In this particular case, there is rearrangement of segments within a chromosome. Now, what is the sequence of segments before mutation? A, B, C, D, centromere, E, F, G, H. Now, observe this. This is not A, B, C, D sequence. It is A, C, B, D. A, C, B, D. Here there is a change in the sequence of segments. Again, instead of EFGH, there is EFHG. There is When there is change in the arrangement of segments within a chromosome, then the mutation is called inversion. If it is apart from the centromere, it is called paracentric inversion. Take this case. A, B, C, D, E, F, G, H. This is the centromere. After mutation, what is the sequence? It is A, B, C, E. That means there is a change in the positions like this, and resulted in A, B, C, E, D, F, G, H. That means there is rearrangement of segments of a chromosome or within a chromosome across the centromere. This is across the centromere. Across. Then it is called pericentric inversion. It's called pericentric inversion. So, the chromosomal mutations can be studied under addition, deletion, translocation, and inversion. And inversion may be pericentric or paracentric. My suggestion to you is, this is one of the important areas for NEET examination. You may have fair chances of getting a question from the types of mutations, especially the gene mutations, the genomatic mutations, and chromosomal mutations.
ఒకటే టాపిక్ సార్ అది దాంతో ఒకటే టాపిక్
జెనెటిక్స్ లో సార్
రేపు కూడా కంటిన్యూ అయ్యింది
సార్